Hello everyone, I'm Helen Patience. In the previous lecture, I introduced you to antimicrobials or antibiotics, where I said these are drugs used to treat infections caused by bacteria. I also talked about the classification of antibiotics. They are divided into four, based on the spectrum of activity, based on the mechanism of action, based on their source, and based on their effect of the bacteria. Effect on the bacteria, where I said bactericidal and bacteriostatic. In today's lecture, I'm going to focus mainly on the mechanism of action. Please, if you missed out on the previous lecture, kindly watch it to help you or to guide you follow up on today's lecture. Don't forget to like and subscribe to our YouTube channel. So, in today's lecture, as I've introduced, I've said we are going to mainly focus on that classification based on mechanism of action. They are divided into 5, where we have inhibition of the cell wall synthesis, disruption of the cell membrane, inhibition of protein synthesis. Then we also have inhibition of metabolic pathways, and also we have inhibition of synthesis of nucleic acid. What are we going to focus on mainly? We're going to focus on how the bacteria is carrying out these activities, such as synthesizing the cell wall, such as protein synthesis, nucleic acid synthesis. We're going to see how the drugs or the antibiotics are going to inhibit these activities. We're also going to look at the mechanism of action, the side effects of the drugs. Cell wall synthesis inhibitors. Let's first understand what is a cell wall. A cell wall is the outside membrane of the bacteria. It's going to protect the bacteria from the external environment. And we also know that bacteria or bacterial cell walls are divided into two. You have the gram-negative and the gram-positive bacteria. For the gram-negative bacteria, these ones, they are having fewer peptidoglycans, while gram-positive bacteria, they are having much peptidoglycan. Also, the gram-negative bacteria, they are the ones that have an outside membrane, while the gram-positive bacteria, they don't have an outside membrane. What do we realize? We realize that both the gram-negative and the gram-positive, they're having peptidoglycan layers. So you want to understand, how do bacteria synthesize that peptidoglycan? That's where we're going to come up with the enzymatic synthesis of the cell wall or peptidoglycan by bacteria. It involves 3 substances. It involves the NAG, and the NAM with amino acids. Where the NAG is the N-acetyl glucamine, and the NAM is N-acetyl muramic acid, and the amino acids. So we want to mainly focus on the enzymatic synthesis, the enzymes that are involved in the synthesis of peptidoglycan by the bacteria. Our first enzyme is going to be MURA-A, also known as N-pyruvate transferase. So the main focus of this MURA-A, it is going to add PEP in the presence of NADP to the compound NAM, to the compound UDP-NAM. I told you NAM means N-acetyl muramic acid, and they are going to form a compound UDP-NAM N-pyruvate. After forming that compound, 3 amino acids are going to be sequentially added. With what are those amino acids? We have the L-alanine, the D-glycine, and the L-lysine to form that compound, also known as the peptidoglycan layer. Other 2 important enzymes that are required in this process are D-alanine racemase, which is responsible for converting this amino acid L-alanine to D-alanine. After converting these amino acids from L-alanine to D-alanine, another enzyme, D-alanine ligase, is going to join up these two amino acids. And lastly, these two amino acids are going to be added to the growing peptidoglycan precursor to form this molecule, which is the growing peptidoglycan precursor. From there, this peptidoglycan precursor, with the help of an enzyme translocase, is going to be added to a lipid carrier. What is a lipid carrier? A lipid carrier, this is a molecule that is going to transport the peptidoglycan precursor from the cytoplasm to the outside of the what? To the outside of the membrane. So this enzyme translocase is going to add the peptidoglycan precursor to the lipid carrier, also known as bactoprenolol. Bactoprenolol, get this word. So after adding it onto the bactoprenolol, other sugars such as N-acetyl glucosamine and 5 amino acids are going to be sequentially added onto the growing peptidoglycan layer, where this bactoprenolol is now going to transport the growing peptidoglycan precursor from the cytoplasm to the outside of the membrane. What happens after it has transported it to the outside of the membrane? It is going to be recycled back into the what? Into the inner membrane by dephosphorylation. A phosphate is going to be removed from it and it will be recycled back to the inner membrane to carry other peptidoglycan precursors. So this peptidoglycan precursor that has been taken into the outside membrane, in the presence of penicillin binding proteins, these penicillin binding proteins are going to aid in the final layer of the peptidoglycan synthesis, which is polymerization. And this polymerization consists of two processes. That is where we are having transglycosylation and transpeptidation. What happens in transglycosylation? In transglycosylation, we are going to see that different sugar molecules that I talked about, the NAM and the NAG, are going to be continuously added onto the what? Onto the growing peptidoglycan layer. They're going to be continuously added onto the growing peptidoglycan layer. As you hear the word glyco means sugars. Sugars are going to be continuously added. Then in transpeptidation, the sugars that were continuously added are going to lie side by side, exposing their amino acids. These amino acids that were added onto each, they're going to lie side by side, exposing those amino acids, where an enzyme transpeptidase is going to create a cross link between the what? Between the amino acids, leading to formation of our peptidoglycan layer, that is the main component in the cell wall of bacteria. So take note of all the enzymes I've written in the red marker. Mira A, D-alanine ligase, D-alanine racemase, and transpeptidase. Now, our main focus is going to be on the classes of drugs, the examples, the mechanism of action, and the side effects. I wrote this earlier to help us save time. I wrote the class, the example, the mechanism of action, and the side effect. Our first class is going to be the non-beta lactams, and they are mainly four. We have the phosphomycin, cycloserine, vancomycin, and bacitracin. What is the mechanism of action of phosphomycin? Phosphomycin is going to block the Mira A enzyme, which helps in the first step of peptidoglycan synthesis. Remember, I told you to take note of the enzyme Mira A. Mira A is going to be blocked by phosphomycin. And remember, MUA is the one that is responsible for adding PEP onto that molecule, UDP in acetylmuramic acid. So it's going to be blocked by the drug phosphomycin. The main side effect is GI upset. GI upset is mainly made up of three answers, that is nausea, vomiting, and diarrhea. Then our next drug is going to be cycloserine. For cycloserine, it's going to block enzymes alanine racemase and dealanine ligase. Remember, I told you alanine racemase is the one that is responsible for converting L-alanine to dealanine, while dealanine ligase is the one responsible for joining the two amino acids, the dealanine amino acids. So it is cycloserine that is going to block those enzymes, and once they are blocked, that means the synthesis of peptidoglycan is going to be blocked. For the side effects, we have depression and peripheral neuropathy. Then the other drug that is commonly asked in questions is vancomycin. They normally ask you, it is 4 marks, describe the mechanism of action of vancomycin. So for vancomycin, for which its mechanism of action, it blocks the processes transglycosylation and transpeptidation. Where I said transglycosylation, glycosylation means adding sugars together, sugar molecules together, and transpeptidation, they are creating cross linkages between the amino acids. The main side effects, we have hypertension, fever, and shock. Then for bacitracin, that word I told you to get, the bactoprenol or the lipid carrier. It is this bacitracin that is going to bind onto the bactoprenol and prevent its dephosphorylation, thus inhibiting lipid carrier recycling, leading to cell death. The side effects, we have GIT upset and have skin irritation. Then the next classes of drug is beta lactams. This is very common. In beta lactams, that's where we are having our penicillins, cephalosporins, carbapenems, and monobactams. So what is the mechanism of action of these beta lactams? These beta lactams, they're the ones that inhibit an enzyme transpeptidase, which aids in cross-linking of the amino acids in transpeptidation, a process called a transpeptidation process, thus leading to cell death. Remember, I told you this is the last enzyme in the synthesis of the peptidoglycan layer. It is the one that is capable of creating those cross-linkages between the amino acids. The main side effects, we have allergic reactions, GIT upset, and super infections. Another important note that I cannot leave behind, we very well know that beta lactams are made of a ring. They have a ring. They have a beta lactam ring. This beta lactam ring is so exposed in penicillins and cephalosporins. That's why bacteria have developed, and bacteria are capable of resisting these two classes of drugs, the penicillins and the cephalosporins, because their beta lactam ring is exposed. So the bacteria produce an enzyme called beta lactamase. It is this enzyme that is going to break down this beta lactam ring. So with time, scientists came up with drugs to help inhibit or kill this beta lactamase enzyme that destroys the beta lactam ring. And some of those drugs called the beta lactamase inhibitors include: 1, We have the avibactam, we have sulbactam. Then we have clavulanic acid, and many other drugs that I've not mentioned. Another important thing to note, except carbapenems and monobactams, for them they are not given out with these beta-lactamase inhibitors. They're not given out with these beta-lactamase inhibitors. Why? That question is commonly asked. They normally ask, why are carbapenems and monobactams not given with the beta-lactamase enzymes? The reason is these carbapenems and monobactams, they have a beta-developed beta-lactam ring. For them, their beta-lactam ring is not exposed. It's surrounded by other molecules around them. So it's protected from the beta-lactamase enzyme. The beta-lactamase enzyme cannot easily destroy them. Also, another important thing to note, these drugs, the penicillins, the cephalosporins, the carbapenems, and monobactams, for them they are further divided into other drugs, into other classes of antibiotics. Please pause and watch those drugs. Understand them well because they are commonly asked in objectives. Okay, next in line we are going to discuss protein synthesis inhibitors. So we have to first understand what is protein synthesis within a bacteria. Protein synthesis is the process by which genetic material is converted into different proteins. How does it occur? It occurs in two processes. It occurs in a process known as transcription and translation. It occurs in two processes where one is transcription and the other two is translation. So what is transcription? Transcription is the process by which genetic material, which is DNA, is converted into messenger RNA. So for transcription, we are converting DNA in the nucleus to messenger RNA. This is A-level biology. I expect all of us to, by this time, to be understanding it. Then, translation is the process by which messenger RNA is converted into proteins. Messenger RNA is converted to proteins. Our main focus today in these drugs responsible for protein synthesis, we are going to look at specifically translation. So, in translation, there are different substances that are needed. In translation, we need three substances. We need messenger RNA, we need ribosomes, and also we need transfer RNA. So what happens in translation? In translation, this is a process that occurs within the cytoplasm of the bacteria. So the messenger RNA that is formed within the nucleus is going to move out of the nucleus, travel to the cytoplasm where there are ribosomes and transfer RNAs. So now let's discuss the process of translation. How does translation occur? Translation occurs like this. This messenger RNA travels to the cytoplasm. Messenger RNA travels to the cytoplasm. We have to first know what are some of the contents that are found onto the ribosome. And the ribosome, we are having codons. These are our codons. These are known as codons. So what happens? within the nucleus, when this messenger RNA comes into the nucleus, the ribosomes, the ribosomes which are divided into two units, that is very important. Ribosome is divided into two units where it's having the 50S, ribosomal subunit, which is the largest, and the 30S ribosomal subunit. Two units, 50S and 30S ribosomal subunit. So once the messenger RNA is within the cytoplasm, the 30S ribosomal subunit is going to come and join onto the 3 codons, onto the 6 codons. Then this 50S ribosomal subunit, which is the larger subunit, it's also going to come and join onto the what? Into the upper part of the messenger RNA. Then another substance, which is the transfer RNA. The transfer RNA is always like this, and it's always carrying an amino acid. It is always carrying an amino acid, and it's having other components also known as the what? The anticodons. Now these anticodons are the ones that are continuously going to pair up with the codons onto the what? Onto the messenger RNA. So what is going to happen next? What is going to happen next is that once the 30S and the 50S, this is the 50S and the 30S ribosomal subunits, once they have joined, the transfer RNA is going to come and join onto the what? And join onto the transfer onto the 50S ribosomal subunit, like this. And base pairing is going to occur. Base pairing is going to occur within the codons of the messenger RNA and the anticodons of the transfer RNA. After that has occurred, remember each transfer RNA is having an amino acid. So it is this 50S that is responsible for it's having an enzyme, peptidyl transferase, that is responsible for creating a peptide bond within the what? Within the amino acids. So once that peptide bond has been formed, this transfer RNA is going to go back into the what? Into the cytoplasm. It's going to go back into the cytoplasm where it's going to pick other amino acids and bring them onto the messenger RNA. So after a peptide bond also has been formed again, this 30S ribosomal subunit is going to come off, and the 50S is also going to move away, and they'll shift to the next codons. They'll shift to the next codons. This is the 50S, this is the under 30S. They'll shift to the next codons. Once they've shifted to the next codons, as they shift, they take along this transfer RNA. As they shift, they take along these codons. So once it has shifted, other transfer RNA are going to come and join onto the anticodons are going to, there's going to be complementary pairing between the codons of the messenger RNA and the anticodons of the transfer RNA. And the 50S ribosomal subunit is still going to create a peptide bond. This process is going to continue until a long chain of amino acids is formed. And a long chain of amino acids that is formed, it's the one that is going to finally coil is the one that is going to finally coil, leading to the formation of the proteins. So our main focus today is going to be about the two classes of drugs. We're having the 50S, the 50S ribosomal, the 50S ribo, sorry, the 50S ribosomal subunity inhibitors, and the 30S ribosomal subunit inhibitors. Let's take these drugs down earlier to save time. So these are our classes of drugs. They are divided into two, where we are having the 30S ribosomal subunit inhibitors and the 50S ribosomal subunit inhibitors. Within the 30S, that is where we are having aminoglycosides, tetracyclines, and glycocyclines. Within the aminoglycosides, that is where we are having examples like amikacin, we are having neomycin, we are having gentamicin, we are having streptomycin. Now our main focus is the mechanism of action. What is the mechanism of action of aminoglycosides? These aminoglycosides, they bind irreversibly, that is the key word, irreversibly, to the 30S ribosomal subunit and block formation of the initiation complex. Remember when we were talking, we said it is the 30S ribosomal subunit that has to first join onto the messenger RNA, then also the 50S joins. So once these aminoglycosides have blocked this 30S from binding, that means the complex won't be what? Won't be formed, thus preventing protein synthesis from occurring. What are some of the side effects? Some of the side effects we are having nephrotoxicity, teratogenicity, and neurotoxicity. Our next class of drugs would be in the 30S ribosomal subunit are the tetracyclines. Tetracyclines are very common drugs and mainly are used for allergic reactions. Tetracyclines and glycocyclines, where we are having doxycyclines, minocyclines, and tetracyclines. What is their mechanism of action? These drugs, for them, they bind reversibly. You see, the keyword here was irreversible. Here the keyword is reversibly. They can ask you for the difference between aminoglycosides and tetracyclines based on the mechanism of action. Based on the mechanism of action, these ones bind irreversibly, these ones bind reversibly to the 30S ribosomal subunit and block attachment of the transfer RNA. For them, when they bind here onto the 30S ribosomal subunit, they're going to prevent the binding of the what? Of the transfer RNA to the S side of the messenger RNA, thus stop protein synthesis. Very easy things. Our side effects, we are having tooth discoloration, we are having photosensitivity, and we are having GIT upset. In the previous lecture, I also told you GIT upset is divided into three: nausea, vomiting, diarrhea. Just in case you've lost the other answers, you can put that. Then we are having the 50S ribosomal subunit inhibitors. These drugs, for them, they are going to inhibit only the 50S. After the 30S has joined, there is the bigger subunit, which is the 50S. They're going to be attacking this 50S ribosomal subunit. And some of the examples we are having phenicals, of which the example is chloramphenicol. The mechanism of action is they bind reversibly to the 50S subunit and peptidase enzyme, which helps in the formation of the peptide bond between the amino acids. What does this mean? This means that when these phenicals bind to the 50S ribosomal subunit, what are they going to do? and they bind to the 50S ribosomal subunit. I told you this 50S is the one responsible for forming. It is the one responsible for forming a peptide bond. Let's say this is our transfer RNA. This is also our transfer RNA, and they are having amino acids at the top. Now, this bond formed here, the peptide bond within the amino acids, it is carried out by an enzyme, peptidyl transferase. So this 50S, these amphenicals, when they bind to the 50S, they're going to block this enzyme from creating a peptide bond, thus inhibiting protein synthesis. What are the side effects? The side effects, we have gray baby syndrome, we have bone marrow suppression, we have aplastic anemia. Another class of drugs, we have ketolides and macrolides. For the ketolides, the main example is erythromycin. Then that's why I had to divide. Then for the microlides, that's why we're having azithromycin, clarithromycin, and erythromycin. What is the mechanism of action? The mechanism of action is that they bind reversibly to the 50S ribosomal subunit, blocking translation and movement of the transfer RNA. Now, for these ketolides and macrolides, for them, they are going to block the movement of this transfer RNA onto other codons. You get? They're going to block the movement of this transfer RNA onto the other codons. How will they do that? By blocking this 50S ribosomal subunit from shifting. Remember, once the 50S is not shifted, even the transfer RNA won't shift. What are some of the side effects? We have severe hepatotoxicity, ototoxicity, and GIT upset. Our next example, or our next class of drugs, will be the lincosamides and streptogramins. This one's, that's where we are having clindamycin, vancomycin, and that. They bind reversibly to the 50S ribosomal subunit at a site overlapping with the macrolides, blocking movement of the transfer RNA. Also, these lincosamides and streptogramins, they're also responsible for blocking. Our main concept is blocking the movement of transfer RNA at a site of overlap with the microlides. Some of the side effects would be skin rash, drug interactions, hepatotoxicity. The next class of drugs is oxazolidinones, where we are having linazolide and tildazzolide. What is the mechanism of action? These ones, they target the 50S ribosomal subunit at this site. The site is 23 RNA site, blocking initiation of the 50S and 30S complex. So these ones bind at a specific site called, let's say, 23 RNA, blocking formation of this complex between the 50S and the 30S ribosomal subunit. Our side effects, therefore, are peripheral neuropathy, optic neuropathy, and bone suppression. That was all about the drugs responsible for inhibiting protein synthesis. Next in line, we are going to look at drugs responsible for cell membrane disruption. There are just two types of drugs responsible for disrupting the cell membrane, where we are having the daptomycin and the polymycins. But remember, the cell wall of the bacteria is mainly consisting of phospholipids. So I want to see how is this layer of the phospholipids disrupted. Let's start with the daptomycin. How does the daptomycin disrupt the cell membrane? This daptomycin, in the presence of calcium ions, daptomycin fixes itself into the what? Into the phospholipid layer. After fixing itself into the phospholipid layer, it creates a pore within the what? Within the phospholipid layer. After creating a After creating this pore, now it has created a pore. After creating a pore, there's going to be efflux of potassium ions within the inner membrane, leading to depolarization, and increased depolarization within the inner cell membrane is going to cause or disruption of the inner contents within the cell membrane, leading to cell membrane disruption. If these pores are continuously formed within the cell membrane, it is definitely going to disrupt the cell membrane. So let me write it for you. The mechanism of action. They're saying that daptomycin in the presence of calcium ions, calcium 2 ions, it creates pores. It creates pores within the cell membrane, causing efflux of potassium ions within membrane, leading to depolarization, thus disruption of the cell membrane. That is our mechanism of action of daptomycin. Some of the side effects we have DI upset, and we also have myopathy. Next in line, we are going to look at the polymycins. So these polymycins, for them, they are having polysaccharides on their membrane. So what is going to happen? These polymycins are going to bind onto the polysaccharides. Remember, within the cell membrane, they are having magnesium ions and calcium ions. So what is going to happen? Once these polymycins have bound onto the lipopolysaccharides, they are going to destabilize the phospholipid layer, allowing penetration of the polymycins, leading to disruption of the cell membrane. So let's write the mechanism of action. We are going to see that the polymycins bind to the lipopolysaccharides, causing disruption or destabilization of the membrane by destabilization of the membrane by displacing the stabilizing ions, e.g. magnesium. Also, these polymycins, they are capable of penetrating into the what? Into the phospholipid layer. Different polymycins breaking down the phospholipid layer, causing its disruption. And that is all about the drugs that are responsible for disrupting the cell membrane. So our next classes of drug is going to be nucleic acid synthesis inhibitors, and it is all about converting DNA to messenger RNA, which we also called transcription. Transcription is converting DNA to messenger RNA. How does it happen? It happens with the help of three main enzymes, where we have DNA gyrase, topoisomerase, and RNA polymerase, topoisomerase 4 and RNA polymerase. Okay, so what is the help of each of these three enzymes? What we are going to see that within the nucleus, before moving out to the cell wall and cytoplasm, but within the nucleus, we are having our genetic material. So now this genetic material is exposed or unwound by DNA gyrase. DNA gyrase is the one responsible for exposing or unwinding of the genetic material to expose the hydrogen ions, to expose the hydrogen bonds. So after the DNA gyrase has unwound the DNA to expose the different hydrogen bonds, it is the role of the topoisomerase 4 to come and break down the what? To come and break down the hydrogen bonds. So topoisomerase is responsible for breaking down. Here it is gyrase. Gyrase enzyme is unwinding the DNA. Then topoisomerase is going to split or break down the hydrogen bonds. This is topoisomerase. Topoisomerase 4 is going to split down the hydrogen bonds to form two strands. Two strands within the nucleus. We are still within the nucleus. It's going to form two strands, where it's going to form the leading strand, and the template strand. Now this is where our RNA polymerase comes about. After the topoisomerase has made the leading strand and the template strand, the RNA polymerase comes onto the template strand. And aids. Now this is our template strand. It's having codons. So it is this RNA polymerase that is going to come and add genetic material onto the what? It's going to aid in complementary base pairing. we call the base pairs. We have uracil, we have adenine, we have guanine. So this complementary base pairing is going to be aided by RNA polymerase. After the what? The base pairing has occurred. That is where the formed messenger RNA is going to peel away from the template strand and move to the cytoplasm. What was our main focus? Our main focus was understanding how each of these enzymes works within transcription. So now let's look at the classes of drugs responsible for inhibition of nucleic acid synthesis. The process that we have just talked about is the one that is responsible for producing the nucleic acid or the DNA and RNA, but specifically the RNA. Now, the classes of drugs, there are two. We are having the fluoroquinolones and rifamycins. Let's start with the fluoroquinolones. For the fluoroquinolones, the examples we have ciprofloxacin, we have levofloxacin, we have mifloxacin and ofloxacin. What is the mechanism of action? These examples of drugs are going to target and destroy the DNA gyrase. They are going to target the enzyme that is responsible for unwinding of the DNA, and they are going to also destroy an enzyme topoisomerase 4. Where we say topoisomerase 4 is the one responsible for breaking down the hydrogen bonds, splitting into the leading strand and the template strand. The DNA gyrase that is going to be blocked is going to be in the gram-positive bacteria, and the topoisomerase that is blocked is going to be in the gram-negative bacteria, thus blocking formation of messenger RNA or nucleic acids. That is our mechanism of action of fluoroquinolones. Then the side effects. The side effects, that's where we are going to be having dizziness, headache, GIT upset. GIT upset is having nausea, vomiting, diarrhea. Then rifamycins. Rifamycins, the examples we have rifampicin, rifampicin. What is their mechanism of action? For them, they are going to inhibit RNA polymerase. That makes RNA from messenger RNA. It can just be even RNA from DNA, as we said here. The function of RNA polymerase, it is the one responsible for forming messenger RNA or complementary base pairing. And that's all we have to discuss about nucleic acid synthesis inhibitors. So our lecture today is going to be about drugs that are responsible for inhibiting metabolic pathways. I said the metabolic pathway is all about synthesis of folic acid, of which this folic acid is very important to the bacteria to form RNA or DNA. So how do we synthesize folic acid? This is the process for synthesizing folic acid. Process for folic acid synthesis. Within the bacteria. How does it happen? So within the bacteria produces PABA, which binds to piperidine. In the presence of an enzyme, dihydropytyrate synthase, we are going to form a compound called dihydropytyric acid, where glutamate is added to form dihydrofolic acid. Now, this dihydrofolic acid is converted to tetrahydrofolic acid with the help of an enzyme dihydrofolate reductase. Our main two enzymes are dihydrofolate reductase and dihydropyruvate synthase. This dihydrofolate reductase is going to help to convert dihydrofolic acid to tetrahydrofolic acid. This tetrahydrofolic acid is a very important acid required for most of the metabolic processes, especially formation of folic acid. So what are the drugs that are responsible for the inhibition, drugs or antibiotics that are responsible for the inhibition of metabolic pathways? That is why we have 1, sulfonamides, and 2, we have trimethoprim. The first drug is sulfonamides. Some of the examples we have sulfamethoxazole and sulfadiazine. What is the mechanism of action of these drugs? These drugs, the sulfonamides, they are the ones that are going to competitively inhibit dihydropyruvate synthase. So these ones, they competitively inhibit dihydropyruvate synthase, which is responsible for converting pyridine in the presence of PABA to dihydropyruvic acid. Some of the side effects we have rash, we have hepatotoxicity, we have bone marrow suppression. Then another class of drug is trimethoprim. This trimethoprim drug is the one responsible for inhibiting. So it inhibits dihydrofolate reductase, which is responsible for converting dihydrofolic acid to tetradihydrofolic acid, thus preventing synthesis of folic acid within the bacteria. Some of the side effects, that is why we are having rash and GIT upset. Another important concept that I don't want to forget, cotrimoxazole is a combination of both trimethoprim and a class of drug, and an example of this class of drug called sulfamethoxazole, whereby if you're given this combination, that means by this sulfamethoxazole is going to be inhibiting the reductase, the synthase, while the cotrimoxazole is going to be inhibiting dihydrofolate reductase. After this lecture, I'm going to display questions that are about antimicrobials to help you understand more about this topic. Thank you for watching. Don't forget to like and subscribe to our YouTube channel. I remain Helen Patience, your farm technician.